Hi, so my name is David Lynch. I'm a neurologist in London at the National Hospital for Neurology at Queen's Square. I'm originally Irish, if you're confused about my accent, although I live in London. So today I'm going to talk to you about what clinical signs and epidemiology tell us about ALSP pathophysiology, that is the underlying cause of ALSP. In translational medicine, we traditionally talk about things going from the bench to the bedside, but the reverse can be very helpful too. There are lots of things that patients teach us about the underlying disease or about underlying neuroscience concepts. So I'm going to talk to you first about the historical perspective on ALSP, then move on to the genetics, what we can learn from imaging of patients with ALSP. I'm going to touch on ALSP caused by genes that are not CSF1R, and then finally what epidemiology teaches us. So the first reports of what we would now recognize as ALSP came out in 1936 in this French article. This was a very large Belgian family published. And although this is in French, I think many neurologists would still recognize this as a kind of classical description of the symptoms those with ALSP experience. They describe a psychiatric syndrome, so predominantly a problem with mood, which evolved rapidly into problems with thinking, which they described as serious and without remission, as well as weakness, followed by stiffness and spasticity. But it was in 1984 when what we could consider the more modern era of ALSP research began with this publication from Sweden. Axelsson et al., they felt they were describing a new syndrome, although they weren't, and they described their new syndrome as HDLS. This was the term that we used for many years. They followed a very large family of more than 71 relatives over 4 generations. And even in that paper, they described a heavy accumulation of microglial cells and questioned what is the significance of this microglial reaction. So even in the very first descriptions of what we call ALSP in the pre-modern genomic era, when the gene was not identified, it was clear that microglia were important somehow. Between 1984 and 2011, a number of individuals and families were described with what was called POLD, or pigmentary orthochromatic leukodystrophy, and HDLS, which is what the Swedes described. But it became clear over time that POLD and HDLS were the same thing. They were one disease, as suggested by Dr. Zoleg even back in around 2009, 2010. And the reason that it was clear that POLD and HDLS were the same thing was that they shared very similar symptoms, so depression, behavioral changes, memory problems, and very similar neurological signs like Parkinsonism, seizures, balance problems like ataxia. And then what really clinched it was that the pathology, when you looked down the microscope of both conditions, was essentially the same. So loss of myelin, which is the lining of the nerves, loss of the axon, center of the nerve, swellings or spheroids in axons, and then pigmented macrophages or microglia. So this is what you see when you look down the microscope. So on the left panel is a patient of mine who had a brain biopsy. So in the top right, you can see these big round brown blobs. These are the spheroids, which are full of abnormal proteins, like amyloid precursor protein. And on the right, on the top right, here you can see these abnormal microglia. And then on this panel on the right side of the slide, this is actually pathology specimens from the original 1936 case. In the top, the arrows point to the abnormal microglia, and in the bottom panel, those black arrowheads point to the spheroids. But the big breakthrough came in 2011 when the gene was identified due to work of a very large international collaboration led by Dr. Zoleg, and it became clear that the cause of POLD and HDLS were mutations in the gene CSF1R. So very quickly, this was replicated by many other groups around the world so that we became very certain that this was the correct finding and that CSF1R is the underlying cause of ALSP. We've seen this slide a little bit before. So you know that microglia are very important immune cells of the brain, and that CSF1R sits on the surface of the microglia. And when it's stimulated, it cascades through a number of pathways that ultimately result in the microglial surviving and being healthy and growing. And another very important receptor that sits on the surface of microglia is called TREM2, and TREM2 has been implicated in risk of Alzheimer's disease as well as causing a different type of leukodystrophy, which is called Nasu-Hakola syndrome. So this is a picture of the CSF1R gene. The vast majority of mutations are in a region of the gene called the tyrosine kinase domain, which is very important for its function. And as of today, we have about 300 patients published in the literature, but we know that the real number of patients affected by ALSP is much more than that, and at least 106 different mutations across the CSF1R gene. So what we see in imaging is we see that this is a white matter problem. In the brain, we have gray matter and white matter. It's predominantly a white matter problem, and it predominantly affects the cortex, the cerebral cortex, especially the frontal lobes, and the frontal lobes are what's responsible for those behavioral changes, problems with planning, memory. And there are parts of the brain that are relatively unaffected, like the cerebellum or the brainstem. And the reason for that likely is because there are different distribution. You have different numbers of microglia in different brain regions. We seem to have a lot more microglia in the cortex, especially in the frontal lobes, and far fewer microglia in the cerebellum and spinal cord, which perhaps explains why they're less affected in this condition. Another finding on imaging that we see in ALSP that we've known about for a number of years are calcifications. So what this white arrow is pointing to are these round white dots. That's an accumulation of calcium in the brain. And this can be a relatively specific sign in ALSP, so that can be quite helpful when we're trying to make a diagnosis. A related but distinct disease caused by mutations in TREM2, which is that other surface receptor on the microglia, cause a condition called Nasu-Hakola syndrome, which is even rarer than ALSP, if you can believe that. And they also, patients with Nasu-Hakola syndrome also have calcification in a different brain region. You can see here this white arrowhead pointing to the calcium. And it was only recently that it was discovered that an explanation for this is that microglia actually have a very important role in regulating calcification of blood vessels, and they do that through TREM2. So these graphs just show mice that have been fed a compound that inhibits CSF1R, so blocks CSF1R, and they have far more calcium accumulating here in the left panel. And then on the right panel, we see mice that have been bred to not express TREM2 properly, and they also have a lot more calcification happening. So that was an interesting link that we saw from the imaging back to the basic science of microglial biology. Another relatively specific sign that we see in ALSP are these unusual diffusion abnormalities. So diffusion-weighted imaging is a type of MRI. And Jen mentioned this earlier in her talk, that sometimes this leads to misdiagnosis because diffusion-weighted abnormalities are more commonly seen in stroke. And so many neurologists who aren't familiar with ALSP might see a scan like this and think this patient had had a number of strokes. If we look, this is an interesting photo from the literature I found of a diffusion scan taken from the side, which is very unusual. You don't normally do that. And you can see that the bright white area here, shown by the double arrowhead, shows that this is tracking along the length of the nerve cell. And what it probably represents is changes in the water content in the myelin, or the lining of the nerve cell, as it's damaged. And these tend to accumulate in ALSP, and that's different to what you see in stroke. In stroke, these abnormalities would go after a couple of weeks and disappear, but in ALSP, they tend to accumulate and persist over a long time. And the only thing that seems to reverse them are the few reports of successful bone marrow transplant, where this diffusion abnormality has seemed to reduce over time. So one question that sometimes comes up is CSF1R the only gene that can cause ALSP? Because in the literature, there have been published some families and individuals who looked very much like they had classical symptoms of ALSP, but we could not find a CSF1R mutation. And that included that original 1984 very large Swedish family that sort of began the modern era of ALSP research. In 2015, I was referred a very difficult case. I'm going to just talk through this patient's history a little bit. So this was a lady who had a normal childhood. She had no family history. She did have a medical history of ovarian failure in her 20s, which we didn't really think was very significant at the time. And then in her 40s, started to develop symptoms with anxiety that was new, difficulty with calculations. So she had difficulty in work. The following year, she developed balance problems, so ataxia. Then she developed a weak and stiff arm and leg, and problems using that arm and apraxia. And this seemed to evolve over a couple of years, like the way you would expect in ALSP. And when we looked at her imaging, we saw the changes that you would see in an ALSP patient. So on the left panel, we see this bright white signal in the white matter. In the middle panel, we see these dots on the diffusion-weighted imaging, as I've just shown you, which we thought were quite specific for ALSP. And this patient actually went on to have a brain biopsy because the local team were worried they were missing something and they wanted to do everything they could to try and make a diagnosis. And the brain biopsy showed axonal spheroids, which you can see in the left panel, these big brown round blobs, and on the right, pigmented microglia. And so the pathologist who looked at this biopsy said, this is an open and shut case, this is ALSP. This is classical symptoms, classical imaging, and a classical brain biopsy of ALSP. And they sent me DNA to sequence and could not find a CSF1R mutation. We thought we'd made a mistake. We repeated the sequencing several times, but could not find a CSF1R mutation. So then we did a more advanced type of sequencing at the time, which looked at all of that patient's genes. That's called exome sequencing. And in that, we found that she carried some mutations in a different gene called AARS2. So when we looked back at a group of patients we had who looked like they had ALSP, but we could not find CSF1R mutations, we found quite a number of people who did have mutations in AARS2 that seemed to be causing a condition that looked very much like ALSP. And this has been replicated by some other teams around the world, including this lower paper, which also had pathology. And we often don't have pathology these days because we try not to do brain biopsies unless they're essential. So if we compare CSF1R and AARS2, the symptoms are very similar. So psychiatric symptoms, problems with memory, so cognitive decline, Parkinsonism, balance problems like ataxia, seizures. The inheritance pattern is different. So we know that ALSP is autosomal dominant, but the inheritance pattern of AARS2 is called autosomal recessive. The imaging appearance is very similar with these diffusion abnormalities that I showed you earlier, which we thought were quite specific for ALSP. And then the pathology seems to be identical in the small number of cases that we have. So the question arises as to why patients with an AARS2 mutation should look similar to patients who have CSF1R. And it's very hard to explain why, actually. So just to get a little bit technical and scientific now for a second, what AARS2 does is it helps to load an amino acid called alanine onto RNA. And amino acids are the building blocks of proteins. So AARS2 has a job in producing proteins, translating proteins in the mitochondria, which is part of the cell that produces energy. And AARS2 is found in all cell types. So this is not specific to microglia. So it's very hard to see why the symptoms and the signs and the pathology of these two completely different genes that have completely different functions would be the same. We know that these group of genes, so I said RS2 is a tRNA synthetase. It puts an amino acid onto RNA. So we know that they can have what we call canonical roles, that is their sort of traditional role in making proteins, protein translation, but they can also have what's called moonlighting roles. So akin to someone having a job and then moonlighting in another completely different job. So those moonlighting jobs can be completely unrelated to their original role. And we don't know whether AARS2 has a moonlighting role in microglia that we don't know about. And that's something that needs a lot more study. And then in 2019, the original Swedish HDLS family was solved. As I mentioned, there was no CSF1R mutation found in that original very large family published in 1984. And in fact, what was identified in that very large Swedish family was a mutation in a very similar but different gene, AARS or AARS1. So we have AARS2 in the patients I described, and AARS or AARS1, just to give it a different name, in the Swedish family. They're both tRNA synthetase, which means they both put alanine onto tRNA. So they have that in similarity, but they're in two different places. So RS2 is in mitochondria. RS1 is in the cytosol of cells. They're different places, but they do similar jobs. One interesting aspect of the pathology here in RS1 has previously been shown that when RS1 is not working very well, instead of putting alanine onto an RNA, it can misplace serine in that place. So serine is a different amino acid, but it's similar enough to alanine that the enzyme gets confused and might put serine on in place of alanine. And that means when you're building your protein, which has all these amino acid blocks, you're going to have some serines in place of alanines. And that means that the protein is made wrong, and so it can misfold and not work properly. And it's possible that this is happening specifically in microglia, and that's why then you have the similar symptoms and signs and imaging and radiology and pathology that you have in CSF1R. So it's possible that this is happening in microglia, but that hasn't been shown yet, and that's also something that needs a lot more research. This sort of hypothesis or suggestion might also explain why patients with RS or RS2 mutations have onset of symptoms as an adult rather than as a child, because if you had a very severe problem making proteins in the first place, then you're likely to have a very severe type of disease that would give you onset as a baby or as a young child. But that's not what happens here. This is an accumulation of a problem that seems to get worse over the course of the life. So just to touch a little bit on nomenclature or naming, so as ALSP can be caused by mutations in CSF1R or RS2 or RS, there has been a move for us to change the naming and to use gene names to be more specific so that we're clear about what we're talking about. We should say that more than 95% of ALSP is caused by mutations in CSF1R. RS and RS2 are extremely rare. But if we want to be very specific, then we probably should use the term CSF1R-related leukoencephalopathy. But there have been a number of name changes in this field already, and I think we need to be very sure, if we decide to change the name again, that we're not doing it lightly and that it won't be just changed again in future. So for the rest of this talk, I will continue to refer to this as ALSP rather than CRL. What do we know about how common ALSP is? That's the epidemiology. How common is it? We know it's a very rare condition. We know that, but how rare is it? Well, really, we don't know. It's essentially unknown. We've done some studies before that show that it accounts for somewhere between 10% or up to 25% of adult inherited white matter disorders. So it's rare, but in some respects, it's one of the more common rare things. So that can be confusing. So this is some data from my group. So this is looking back at our last sort of 70 or 80 diagnoses of patients with white matter disorders. And by far and away, CSF1R was the most common diagnosis we made. That's very similar to Jen's work. I'll say that we don't have ALD in this group because I don't see ALD. ALD is seen by our metabolic team, so I don't make ALD is second on that list, but I don't make diagnoses of ALD. So CSF1R is certainly one of the more common disorders that we see. But knowing that something is common amongst a rare condition like white matter disorders, it still doesn't tell you anything about the population, how many patients are out there, how many patients are in the UK, how many patients are in the US. We don't know. So one effort that we've made to try to work out that number is by looking at something called UK Biobank, which is this very large database that exists in the UK, and it recruited 500,000 people to take part in a study between 2006 and 2010. And all of those people, 500,000 people, gave lots and lots of blood samples. Some of them had lots of scans and imaging done, and they all linked their primary care data, their hospital records. Some of them had tests of their memory. So this is an amazing resource that we have that can look at the population. For about 470,000, or nearly all of the people in it, so almost 500,000 people, we have very detailed genetic information, which is what we call whole exome sequencing. And then, as I said, we have linked health records, both primary care, hospital records, ICD-10 codes, and what are called algorithmically defined diagnoses. So what we did is we pulled all of the rare variants in CSF1R out of those nearly 500,000 individuals. And then we looked at these mutations, these variants, and we classified them using criteria called the American College of Medical Genetics criteria, ACMG criteria. And we could say that they're either pathogenic or likely pathogenic, or the term that many of us dislike, a variant of uncertain significance, a VUS. We know that most mutations that cause CSF1R are in the tyrosine kinase domain, so in this particular part of the gene, and often they are classified by these criteria as a VUS, which can cause a lot of confusion and difficulty for families who receive that diagnosis. So looking at the numbers that we found, so we found 25 individuals in biobank with a pathogenic variant, more with what we call a likely pathogenic variant, so 107, giving us a total of about 28 people per 100,000, or 1 in 3,500 people carrying what we think is as a damaging mutation in CSF1R. And that number is many, many-fold higher than you would expect for a disease that has 300 cases published worldwide. So there are far more people in the population carrying a damaging mutation in CSF1R than you would expect. So I mentioned that most mutations in CSF1R are in the tyrosine kinase domain, and often they don't quite meet the criteria by the ACMG to be called pathogenic or likely pathogenic. They're often called a VUS. And so we still wanted to look at those because we do think many of them do cause ALSP. We know that for sure in the clinic. And looking at that, we found 20 variants in 50 individuals. So if we're going to include those, and we think some of those are pathogenic, then the frequency of a damaging mutation in CSF1R is about 1 in 2,500 people, which again, I think is far higher than you would expect. We then looked at what were the clinical outcomes. So what were the implications of having a variant in CSF1R? So we compared individuals carrying mutations that were pathogenic or likely pathogenic to a group of 20,000 random controls, or healthy people who were in biobank. And we looked at their self-reported diagnoses, their ICD-10 codes, what they call algorithmically defined diagnoses. And we found that if you had a pathogenic variant, you were 5 times more likely to have a psychiatric code in your chart. You were 10 times more likely to have a depression code in your chart. You were 20 times more likely to have a self-reported Parkinson's disease code in your chart. And combined pathogenic and likely pathogenic group had increased risk of algorithmically defined dementia and 5 times risk of vascular dementia. So what this is telling us, and this is limited because this is biobank data recruited from 2006 to 2010. It has followed on from then, but some of this is from the pre-CSF1R gene identification. Maybe some of these people actually have ALSP or have early ALSP. So what we've taken from the biobank is that CSF1R variants are far more common than expected in the population. And if we were to extrapolate that over to the world, there'd be more than 2 million people in the world who carry a pathogenic or likely pathogenic variant, with the warning there that this is UK data, so it might not be applicable to other populations. We've shown that CSF1R variants are associated with both neurological and psychiatric diagnoses, so that might represent early or misdiagnosed ALSP in those individuals from biobank. So that question arises, we see far more people carrying a variant than we see manifesting the disease. The question is this unusual? Do we see this in other genetic disorders? And the answer to that is yes. So this slide is data from a colleague of mine at UCL, Arianna Tucci, who looks at completely different genetic disorders caused by expansions, what are called repeat expansions, and those are conditions that are things like Huntington's disease or ALS caused by a particular expansion called C9orf. We know a lot more about the frequency of Huntington's disease because it's so common. So we have quite accurate information about how common it is. And what these graphs show is the light blue lower peaks are the expected frequency of the genetic disease, and then the very large dark blue graph is the frequency of the repeat expansion in the population. So essentially what she's shown is that there are almost 3 times the number of people in the population carrying a repeat expansion that would cause Huntington's disease or ALS than you would expect. So this finding is not unique to ALSP. This is what we're finding in lots and lots of other genetic diseases, that the number of people in the population carrying these potentially damaging variants is much higher than we expected. So the question arises, why is that? So we can think of it as like the tip of the iceberg. There are maybe two explanations that don't have to compete with each other. Maybe there's a bit of both of them being true. One is that we might be missing patients who have the disorder, so that the level of the iceberg above the waterline are the patients that we identify as having ALSP and receive a correct diagnosis, but that there are many, many more people in the population who have ALSP, but it is never recognized, they're misdiagnosed, they can't access genetic testing, or that they have a very mild or they present very late. The second possible explanation is what we call reduced penetrance. So penetrance is the relationship between having the genetic mutation and having the disease. If everyone who has a genetic mutation gets the disease, you would say it had 100% penetrance. If half the people who had the mutation developed the disease, you'd say it had 50% penetrance. So there might be need for what we call a second hit, so that you have the gene mutation, but then you need to have some other factor that then causes the disease to develop. We don't know what the penetrance of ALSP is. We know that there are previously published estimates that are very high. So some people come to me saying, I've read that the penetrance of CSF1R is 95% by age 60, but those early studies are very biased because they were constructed using families that had many affected family members. And when you do that, you get information about those families, but that doesn't necessarily represent individuals where they're the only person in their family who has ALSP. So there's some biases in there that affect our understanding, and I think we need to do some large-scale studies to try to determine the true penetrance so that we can genetically counsel people accurately when they have a predictive test. Some of the factors that might influence penetrance, and these are just speculation, so I wouldn't make too much of this. We know that head injury has been reported to precede symptoms in some forms of adrenoleukodystrophy and in CSF1R. We know that some viruses are important in neurologic disease, like Epstein-Barr virus is very central to the development of multiple sclerosis. So is there some viral trigger that needs to happen? Or is there some regulation by a different gene that somehow influences CSF1R, maybe its function or its expression? And that's been shown in a different type of dementia called frontotemporal dementia. And some of these questions will be answered very nicely by Jen's study, and that's why she's asking these types of questions in her study. So that brings me to the summary. So what I hope I've shown you over the course of this talk is that clinical signs and imaging can help us to understand microglial distribution and function, as nicely shown by recent work on the importance of microglia for calcification in the brain. There's a lot more to be discovered regarding the role of these alanine tRNA synthetases in microglia, and that there's a much larger group of individuals who we would say are at risk for ALSP than previously known, and there must be some genetic or environmental factors that influence risk. And we do need to do further work to understand better, more accurately, the penetrance of CSF1R mutations, so that when someone has a predictive test, we can give them accurate information. So of course, I'd like to acknowledge all the patients and families and caregivers that take part in all our studies and everyone who's come here today to talk. I have to acknowledge my colleagues in the Queen's Square Adult Leukodystrophy Group, especially my clinical fellow, Charlie Wade, who does a lot of the work for me, my clinical nurse specialist, Anna, and some of my mentors. And I'd definitely like to thank you for inviting me here to talk today.